people. So I call myself, I'm actually a marine biologist by training. And biogeochemistry of sea ice is something that quite a lot has been written on about recently. It's got the term biology in there, it's got the term chemistry, and of course both the biology and the chemistry interact to give us the geology of Earth systems. But I'm more of an ecologist than by biology, so I have actually published this term eco-biogeochemistry in one paper. So I managed to do that. I would really love, and I'm edging towards it, to publish the term physio-eco-biogeochemistry. And I guess that it is easily possible to get a new discipline of paleo-physio-eco-biogeochemistry in the not too distant future. And who knows, maybe now we can have astro-paleo-physio-eco-biogeochemistry as well. So we're working on it. My career aim is to get this one published. I'm sure we'll manage that. We'll see whether I can go on long enough to do that. So I'm working in Menai Bridge, which of course you've all heard of, the centre of polar research in the world, which is in Bangor in North Wales. This is about the only homage that we can make to polar research. In 1963, there were some ice flows opposite the pub where we all drink, but actually this is not sea ice, this is not frozen water, seawater, this is actually ice that had formed in the rivers and been washed down from altitude, so it's not even proper sea ice. But anyway, so we're a small group, we're me, a postdoc and a co-worker, but a lot of my work has been together with colleagues in the Alfred Wegener Institute, which is where I did a lot of postdoc work, and colleagues in Finland as well. So, sea ice biology, ecology, here's a little bit of history. Here's definitely an ancient and noble science. Here's a map of Iceland that was drawn in about 1590. And this is a translation of this work by a guy called Hexham, where he talked about these huge and marauders heaps of ice brought hither with the tide from the frozen sea. So here, you can't really see it because of the light, but there's ice flows up in this what is clearly Iceland, making a great and terrible noise. So already we've got the kind of the concept that there's a lot of the stuff. There's the huge amounts of ice flows, and the tides are important, or the ocean currents are important. So we've got the oceanography covered as well, making a great and terrible noise. Some pieces which oftentimes are 40 cubits big. Well, my guess is he's talking about icebergs then. These are massive pieces of ice. And upon these places, white bears do sit closely, so clearly polar bears are sitting on the ice, watching the silly fish which here about do play and sport themselves. Well, so we have the ecology, the polar bears eating the fish that are associated with the ice as well. So it's a noble thing. Ice is important for birds. It's important for the charismatics biology that we all know so well, and the mammals too, the polar bears and everything else. And we had a synopsis from John early on as what it's all about. There's a lot of top-level trophics. This is a small inlet in fast ice in the Antarctic. And if you think that each one of these blobs, which is a Weddell seal, is 2 tons of biomass, and this is the age of a penguin colony that extended back at the same scale as here to several kilometers down the road there. There's an awful lot of biomass in these polar regions, and that's because there's an awful lot of stuff for these things to eat. You can't have a lot of the higher trophic levels in biology if you don't have a lot to eat. And in the Antarctic, John talked about amphipods in the Arctic being eaten by polar cod. In the Antarctic, where the penguins and the seals that I've just shown, basically the food source is this beast. It's a crustacean, grows to about that big. It's called krill, Euphausia superba. And that basically, when a krill swarm swarms, the whole of the sea for square kilometre after square kilometre is turned blood red, because they're just there in their millions. And when, in one mouthful, a humpback whale can take up several tons of krill, and it takes lots of mouthfuls a day. So there's a lot of krill there, and there's a lot of krill there because the krill in turn are feeding on the primary producers. The key players are the bacteria, the photosynthetic organisms, which are unicellular algae, and protozoans as well. So there's an awful lot of krill there, but to support that part of the food chain, you need an awful lot of that stuff as well. So that's basically the biology in a nutshell that we're studying. Now, when John asked me to give these lectures, he was remembering some lectures that I gave last year in the summer school in Svalbard, and he said basically to give the same things again, and that's what I'm actually going to do. So what I'm going to do is the first half, before coffee, is I'm going to go through some of the basic biological concepts, at least as we understand them as marine biologists or marine biogeochemists, and some of the key factors that are at play. And then after coffee, I'll go in and describe how they're pertinent for those organisms that are living in the ice. Now, I suppose I could be talking to myself after coffee, but then there you go. Hopefully one of you comes back. So we've had about 150 years of research into sea ice, about the biology of sea ice, but it's only since the 1970s, 1980s that we've really studied sea ice biology in any detail. There's been a much longer history into the physics of sea ice, spanning back, I suppose because people were interested much earlier how ice got in the way of ships before they were interested in the kind of things that grew in there. But this is an intriguing picture that I kind of put up just to show that Joseph Hooker went down in 1842 on the Erebus and Terror expedition to the Antarctic, and this is one of his sketches from his lab book of some organisms he isolated from the ice. You can see that there's some round ones, there's some long, thin ones, and there's some chains of ones as well. Annette Bartsch, a German colleague who went down in 1990, took this picture down the microscope, and basically it's the same picture. And I just think the symmetry of this, as far as scientific history goes, is really quite nice. You know, this was the first picture to ever be taken of life in the ice, basically, and then, you know, quite significantly later, somebody else comes and takes the identical picture.
What are those things?
I'm going to come back on to what these are, but basically these are unicellular algae. These are photosynthetic organisms. They're called diatoms, and we'll go into why diatoms are important in a little later. But basically they're plant-like organisms that grow in the sea. So here we've got this crash course in marine biogeochemistry. It really is, in its simplest form, supply and demand. Space, light, nutrients. By nutrients I'm going to refer to nitrogen, carbon, oxygen, phosphorus, iron. The list of nutrients that are important for life just goes on for pages and pages and pages, vitamins and everything else. But each one of those nutrients is limiting to growth in some particular way. Grazing, so how much food you can get if you're a grazer, or if you're an organism that's being grazed, how you can stop being eaten by something else. And of course how you disperse yourself to as far and wide as possible. So not just how you disperse the adult phases of your existence, but also how you disperse your progeny and ultimately your gene pool as well. Space generally can be thought of, you know, for an organism that needs to photosynthesize, it needs to be high up in the water column, it needs to be in the lit part of the ocean, it doesn't want to be sitting 6 kilometers down in the ocean where it's completely dark. So space for photosynthesis for synthetic organisms means keeping up there in the light. For a bacteria, it doesn't really care where there's light or not. It's able to reproduce and function without light, but it does need there to be a food source. So of course space for the bacteria is going to be somewhat different. Light is driving everything, and that's what we're going to see now. So space, rather an obvious one. So the general spatial sphere for a bacteria is on the micrometer scale. So, you know, micrometers are important for your bacteria, but the algae and protozoans, so these are the plant-like organisms, and the things that eat them, protozoans. Unicellular organisms in general, mostly. You know, micrometers to centimeters are important in their existence. Crustaceans, so crustaceans, shrimp-like organisms, things with a hard exoskeleton. They extend in size range from, some of them are only a few microns big, through to things like that krill that I showed you that is a few centimeters big. So they, you know, their kind of sphere of influence is centimeters to hundreds of meters. And because I suppose they're wafted with ocean currents, you could even put kilometers in here as well. So it can extend to the high end of this hundreds of meters. Fish, obviously, you know, tens of meters to hundreds of kilometers to thousands of kilometers if you're a salmon or an eel, and whales, or the whole of the globe, basically. But everything from a bacteria cell, so everything, any one of these kind of huge size spectrum, everything ultimately sinks to the bottom of the sea. It dies, it sinks. And so on the vertical scale, sinking particles, the scale is thousands of meters. So I, you know, if you give this lecture to, and mostly undergraduates would come up with this eventually after a lot of pulling of teeth and everything else. You know, they would kind of give you this list after about 6 hours of trying to get it out of them. But this one is intuitively not thought about it, because we always think about the horizontal scales rather than the vertical scales.
What's the vertical scale for life though? Because they settle to the bottom.
They settle to the bottom. So yeah, well, so in the clearest of oceans, light will penetrate. So in there's some, I can't quite remember where it was, some coral reef area somewhere, some very, very clear waters, very crystal clear blue waters. They measured photons of light, and therefore they actually found some seaweeds that were growing still at 263 meters. Now that's the absolute world record of something that is attached to a substrate and still growing. There was enough light down there. But in general, light, you're talking about in the Antarctic, again, beautifully clear waters, 50 meters, and then it's gone.
But this is related to the life depending on the sunlight, because there are other...
Yeah, no, so this is purely, talking about photosynthetic organisms. But yeah, down at, in the very deepest of the ocean, you've got bacteria there that are growing quite happily, so you're down to 13 kilometers. So in the deepest trenches, there's bacteria still. They're very tolerant of high pressure. So light-based life, top 50 meters, say, although extending down to 200 meters in places, but life in general, absolutely everywhere. There's no part of the ocean that isn't colonized. So, but if you kind of, this has become one of the most famous pictures in marine microbiology, and I think it's a con, because it was drawn on the back of an envelope by a drunken professor at a conference, and it got published in Science. I mean, if I drew in a drunken state a cartoon of what I thought life was, I mean, they sent it to Science, they would laugh at me. Well, they wouldn't actually acknowledge my letter. So, but this kind of, it does kind of show some of the spatial scale. So it shows that it's not uniformly distributed. Even, you know, if you say that this is a view down a microscope slide, a low-power microscope slide, you'll see some algae, some phytoplankton. And then you'll see some protozoans that are things that eat the phytoplankton congregated around them. In other places, you'll get a lot of bacteria that are swarming around some decaying matter. And this is already a good picture when we kind of put it into what we think about life in the ice. If we think about life within a block of ice or in a brine channel, it's not going to be uniform. It's going to be different organisms that are going to occupy different parts of that system. And their interactions, you know, this bacteria here is going to have a very different dynamics and experience in its physical and its chemical and biological conditions than the bacteria that are congregated around this particle here. And one of the biggest challenges which we'll go to after coffee is actually trying to understand the dynamics at the kind of the cellular level in a maze of complexity such as this. Okay, think phytoplankton. So these are photosynthetic plant-like organisms called algae that are carried by ocean currents in just about all parts of the world. And people think this kind of term diatoms. So diatoms are a special kind of phytoplankton. What characterizes them is that they are encased in a glass case, if you like, made of silicate, a very rigid cell wall, which I'm going to come to on in a little bit more detail. You'll see in this picture, some of them have spines. This isn't a diatom, actually, this is another group. Is that the earthquake?
No, that's lunch.
That's lunch.
Those are the rocks falling. Yeah.
Amazing, isn't it? And you can see there's different shapes. Some of them are very ornate. Some of them are very small and kind of immersed into some kind of jelly-like substance as well. So there's a huge variety of shapes and forms of these diatoms. But basically they're encased, so the cell is encased in this very rigid silicate wall. And diatoms are a huge part of the CI system. They are by far the most dominating organism that lives in sea ice that we know so far. And that partly that is a result of the rigidity of this glass frustule. So you can see that this glass frustule has holes in it, and this allows gases to exchange, and it allows nutrients to go in and things to pour out. So it's, it's not a closed system, so exchange happens through the pores. But this is a neat piece of work done by some colleagues in Bremerhaven, where they've kind of made virtual models of these silicate frustules, and they've done load testings of just how strong they are. And they've also taken some real things, and they've actually kind of tested them under the microscope by putting different loads of these needles onto the cleaned up frustules and actually seeing how much force is needed to break them. And basically, about 36 nanonewtons are needed to kind of cause this kind of deflection. But basically, this is all well and good, and the papers are fascinating read. But basically, they equate trying to break open a diatom is like us trying to break open a Brazil nut with our back teeth. Well, what's going to happen is our back teeth are going to break before the Brazil nut. So because they've got the silicate frustule, they're able to withstand tremendous physical forces. And of course, that's one of the reasons why they're so successful within the ice. There's an interesting aside. The crustaceans that eat diatoms have been involved, and this is a picture of one of the teeth of a crustacean, and they've got these kind of structures a bit like our teeth that are also made out of silica. And there's been an evolutionary arms race between the diatoms and the crustaceans. So as the diatoms have laid down more and more silica in their frustules and therefore become more and more difficult to eat, the crustaceans have had to adapt their feeding behavior and also their mouth parts to be able to crush them. It's a bit similar to the arms war that there's been between herbivores and grasses. Because in a grass, if you rub your finger down a blade of grass, it feels very rough. And that's because it's full of silica particles. And what the silica particles do is they wear down the teeth of the things that are grazing them. And there's been a similar kind of an arms race between grass species and herbivores and in terrestrial systems as well, but we get carried away. So, but it's not just diatoms. So if you start getting into the sea ice literature, you're going to read diatoms, and you would be excused for thinking that they're the only things that are important. Well, they're not. This is a fluorescent picture of bacteria. You stain them with a dye, and you shine a light on them, and they fluoresce. And they fluoresce blue. So this is, oh sorry, I'm being too loud.
Make a copy and not distribute.
So the blue dots are the bacteria. This is exactly the same picture, just fluoresced in a slightly different way. So this blob there is that blob there. But what you see here is red and yellow dots. I apologize if you're colorblind, because somebody got quite cross with me once because he said, you talk about all these different colored dots, they all look white to me. But anyway, but the red and the yellow dots are photosynthetic phytoplankton as well. So there's bacteria there, but there's bacteria sizes of organisms that are also, they're not bacteria, they're also photosynthetic. Bacteria are not photosynthetic, these ones are. So, and we're only really just in marine science getting a handle on what these small beasts do. We don't really know. More and more literature is being churned out, but still, in the grand scheme of things, my guess is, it's still in the early stages of understanding it. So, of course, I've talked about photosynthesis, and there's a certain group of organisms that are photosynthesizing. All organisms are respiring in one way or another. So the process of photosynthesis in its very, very most primitive form is taking light, visible radiation, and it's converting CO2 and water, producing organic matter. A byproduct of that is oxygen. And then respiration is exactly the opposite. It's breaking down of organic matter, consumes oxygen, and produces CO2 and water. So if you like, in its very most basic form, this is what most of us that are doing in biogeochemistry, this is summed up almost in this one slide. So you're taking light and you're throwing out at the end of the process some heat at the end of the day. Oxygen is present throughout, and basically we're kind of going from, so I'm using this term. I'm using this term as a general kind of term for organic matter. It's obviously a lot more complex than that, but basically this is respiration going on here to produce the CO2, the water, and the energy. The energy that's needed to drive biological processes. So basically, the source of the energy is respiration is kind of very much more the important process. This is very much more the important process than the photosynthesis. You know, the photosynthesis is there to produce the organic matter, and of course to keep the oxygen there. But again, biologists or biogeochemists tend to get caught up on the things that are limiting photosynthetic production, and I already did that in the way I introduced the subject matter to you. Whereas actually, we shouldn't forget the respiration stages of it as well. I hope this is all relevant. Actually, I think it is to understand what I'm going to say next. To just go into a little bit more detail about photosynthesis, in that there are two reactions. The one that you need light for. So basically, this is what you need the light for. So you're actually trapping the energy, and that's where the oxygen is being produced. And then there's the dark reaction, which is where basically the CO2 and the energy that is produced up here and the electrons that are produced actually goes to make the organic matter. So the light part of photosynthesis is only one fraction of the story, if you like. This is what's written in every textbook, this bottom equation here. So some water, some CO2 and some light gives you the organic matter, oxygen and water. But actually, it is important to realize that it's kind of, there is a light part and a dark part. So in the light part, you trap the energy from the light, and then the dark reaction is where you build the organic matter that you're interested in.
But the dark, so
Is chlorophyll the only molecule that?
No. There's chlorophyll is the kind of the dominant one within most systems, but there's a list of many pigments that can kind of develop to make that matter.
And the dark part of the reaction happens at the same time, basically, or?
Yes, it does. Yeah. Well,
Not within the solid.
So when you think about the light reaction in the water, so the light profile in water, you have a lot of light at the surface, and it kind of goes down. And it's not just the amount of light that is important, but it's also the kind of the changing of the spectra. So this is the spectrum of light, general spectrum of light, going from about 350, or from the UV, up to about 700 nanometers. This is what a kind of general spectrum of light looks like at the surface of the water. As you go down with increasing depth, you can see that the spectrum of the light becomes very much narrow. So you know, in this water body, a certain depth has a very narrow spectrum. Now the change in this spectrum and the shape of the spectrum depends what's in the water. If there's a lot of sediment, if there's a lot of humic material, this is a river in South Africa where there's a lot of brown humic material, so this water is going to have a very different spectrum of water with no brown stuff in it as well. So, and of course depends on the depth. This is the spectrum of chlorophyll A, and you can see that within a general spectrum, chlorophyll A traps light at 2 parts of that spectrum. But there's a whole range, and this goes back to your question, there's a whole range of other pigments that photosynthetic organisms have within them that trap light in different parts of that spectrum. So if it was just based on chlorophyll A the green stuff that we know so well, there would be an awful lot, large part of the spectrum that is never ever utilized. So photosynthetic organisms have a host of different pigments that they can change very rapidly. You know, they can build up, you know, so an organism up here, trapped up here, would have a very different pigment composition than an organism trapped down there. Within minutes, the pigment composition can actually change. So during the day, as an organism is kind of being mixed by surface circulation through all of this, this composition is changing as well. So it depends on how deep it's being mixed within the water column. Well, yeah, I think we have to go into this a little bit. Irradiance, so we don't talk about light intensity, we talk about irradiance. So as light energy increases, the amount of photosynthesis goes up as well. So basically, lights until it reaches a maximum where it plateaus off, and you can't saturate the system anymore. It has become saturated. There's some key points here, is that you need a certain amount of light energy to actually compensate for the energy losses due to respiration. So I said respiration is kind of going all of the time. A photosynthetic organism is respiring all of the time. Well, to actually compensate from those energy losses, you need a certain amount of light energy to actually just bring the whole thing to cost neutral. You know, so and these tend to be very low irradiances, but still there needs to be light there for it to happen. If it's below this light level, basically the organism is respiring and therefore is in net energy loss. So a seaweed in winter, where there is no light in a polar region, will always be in this part of the thing. So if it hasn't got enough energy reserves within it to actually to last that long, to respire that energy off, of course it will die. I think so. That's the key point to come out of that. So, I think I'll stress it again, all organisms are respiring. Photosynthesis produces oxygen. So where there's photosynthesis going on, oxygen is being produced, and it consumes carbon dioxide. Respiration is exactly the opposite, consumes oxygen and produces carbon dioxide. So in anything, when we go into the field and we measure levels of oxygen, you know, I go to the sea and I take a bottle and I measure the oxygen in that bottle, that is going to be a reflection of the amount of photosynthesis and respiration that has been going on in the history of that water sample over time. So if there's been a lot of respiration going on, the oxygen values are going to be low. If there's been a lot of photosynthesis, the oxygen values are going to be high. For the biologists here, I apologize for the statement. It's not quite true. There are other organisms out there that contain other things other than chlorophyll that are able to photosynthesize with it. Okay, so you can kind of, I'm not going to go into this, but you can measure with time, you can switch a light on, and you can measure the fluxes, and what you would do is you would see that as the organism is photosynthesizing, oxygen is being produced, respiration is consuming oxygen, so therefore what we're able, you know, basically there's several terms here. You can measure the respiration, you can measure what we would call the net photosynthesis, but actually the total amount of energy within the whole process, the system, is actually a gross photosynthetic term where the two are added together. So this loss term is kind of key. Of course, for there to be net energy gain and therefore growth, this term has to be very much higher than that term there. No, that's not true. I'm not going to talk about it, but basically, it's just interesting is that there's an enzyme there within most photosynthetic organisms called 1,5-ribulose bisphosphate carboxylase oxidase. Basically, 30% of the protein on the planet, or planet Earth, is basically made up of this one enzyme. It's so fundamental to photosynthetic organisms. And basically, this is where the fixation, the conversion of the CO2 into organic matter is actually being kicked in. So basically, through this pumpkin big, which is a very inefficient protein, but anyway, we make the precursing organic matter for a whole range of other things that basically every organism needs: carbohydrates, lipids, proteins, and nucleic acids. We need carbohydrates for structure and energy, lipids for structure and energy reserves, proteins are therefore mainly there as catalysts, and nucleic acids for genetic information and reproduction. And so basically we've gone from light and CO2 and through a whole complex, we can kind of come up with this, which is the same for algae, is the same for most bacteria, is the same for crustaceans, is the same for whales, is the same for humans. You know, we've all got characteristic build-up of these four classes. And this is what you look like of an algal cell. You know, a typical algal cell is 40% carbohydrate, 15% lipid, 40% protein, and a little bit of nucleic acid, 5%. And then you can actually do very easily, you can do the maths and work out, well, you know, how much carbon is needed, how much hydrogen, how much oxygen, nitrogen, phosphorus, and so on. You kind of come up with these kind of ratios. But basically for a marine organism that is 50% carbohydrate, 40% protein, and 10% nucleic acid, which is, you know, is fair enough. Basically you know that there's a need to have carbon, nitrogen, and phosphorus, which are the main nutrients required for growth, and basically this ratio, which is 100 to 15 to 1, which is called the Redfield ratio, and it is a pretty good marker for what's going on. So we know that by looking at the carbon, nitrogen, and phosphorus content of organisms in the sea, we can do quite a lot with this very powerful ratio that we have there, which is based on nothing more than a kind of a breakdown of what a typical carbohydrate is made of.
Sorry, can you go back? Completely naive question.
Good.
You got this by doing the energetics. You did the budget of it.
Well,
Is there someone did.
Yeah.
So is this Redfield ratio? Has anyone tried to calculate this from more first principles, or say that it's changed over evolutionary time, or
Yeah, there is people out there, and I suppose the more recent papers are beginning to be a little bit more critical of the Redfield ratio than I'm kind of putting forward now. But it is remarkable, you know, you dip your bucket into the sea or take a lump of ice or whatever, and you measure carbon, nitrogen, and phosphorus balance, you very often get the number that's very close to this Redfield ratio. So, but there are subtleties in there that are.
When you build this discussion, how about you have organisms which are not based themselves on oxygen, for example, anaerobic?
Yeah, but within the sea ice doesn't exist. Well, no, there are anaerobes in the sea ice, unfortunately. There's a few, but within surface waters of the world's oceans, there's very little anaerobic activity that's going on. So that's why I'm not.
Yeah, okay.
In this general view,
It's almost irrelevant by comparison.
For ice-based systems, it's almost irrelevant. I say that with a little bit of trepidation because I know that there's now 3 papers out there that are talking about anaerobes and sea ice. So, but nobody's really been able to explain how they get there and what they do, to be quite honest. So, yeah. So yeah, that is a. Yeah.
I take it from what you just said, that human activities did not change the ratio significantly up to now.
No.
Because we're dumping lots of phosphorus, for instance, by fertilizers.
No. So this is not the ratio of those elements in the seawater, which we are mucking up in coastal waters. This is the elemental ratio within the organisms themselves. So. You know, if I throw you into a soup of nitrogen-rich water, you're not going to change your elemental composition too much.
But does this suggest something about what the human diet ought to be?
Well, yeah, I mean, we do the same kind of analysis when we look at, you know, diet as well. I mean, yeah, I suppose ultimately we do. We know what the ideal fat content of a human being should be. It should be somebody like me, really, not somebody like John. And you know, we know what the energy requirements are to maintain the ideal skeleton. So yes.
Sorry, maybe this is very obvious, but I don't understand why it should be so hard to explain an anaerobic organism in sea ice.
Because there's not very many parts of sea ice, even in a 12-meter thick, heavily rafted piece of sea ice, there's not much of that area that is actually going to be low in oxygen. If you take, well, I'm going to show some data later, but I mean, but if you go and take a sack hole sample, so basically if you partially drill a core and let the brine trickle in, and so you actually measure the oxygen directly in the brine, you very rarely find low oxygen values. You certainly, there's never been a report, even by the people who have found anaerobes in ice, where there's been zero oxygen. So you quite often find depleted oxygen. Well, actually, you most often find high levels of oxygen and depleted carbon dioxide. Is the way it tends to go. So you normally see, but that could be because we've tended to sample in summer and spring when there's a lot of photosynthetic activity going on, and we don't have people going in the depths of winter to sample ice. But we can come back onto that because I am going to show some oxygen and carbon dioxide plots. There you are. There's a call to bring you in after coffee. Nitrogen is a key limiting beast. Nitrogen is basically, you need nitrogen to make amino acids, and you make the amino acids from the ammonium ion. But nitrogen is available within marine systems, at least, in the forms of nitrate, nitrite, well, urea a little bit, and ammonium. There isn't very much ammonium in seawater. In most parts of the world, there's quite a lot of nitrate, and this comes to what you were saying, in coastal waters, the Baltic Sea in particular, there's humongous amounts of nitrate. But basically, nitrate, before it can be built into an amino acid, has to go through several stages. It has to be converted into nitrite, which is then converted into ammonium, and that costs energy. If the cell is able to take up the ammonium directly, that doesn't cost any energy whatsoever, well, relatively little. So there's a conflict within marine organisms in their nitrogen requirements. Of course, there are amino acids floating around in the sea as well, and they can be taken up directly by photosynthetic organisms and used directly. But again, they're there in very, very low numbers. So there's a conflict in the energy requirements. And in sea ice, and I'll show this data, ammonium can be there in humongous amounts. I mean, really huge amounts that are unknown in any other marine system except some sediment systems. And what happens when ammonium is there in a large amount, what it does is it stops this enzyme, this nitrate reductase enzyme, working, and actually prevents the cells expending the energy on taking up the nitrate. So we do find in the sea ice that this pathway, this is the normal pathway within marine systems, but this pathway can dominate within the sea ice system.
And urea, how much is there?
There's titchy amounts. I mean, even if you go in the middle of a huge, fish swarm with a load of crustaceans in there as well, there's still pretty titchy amounts. It's not a main driver within the system at all.
So it can't rectify the process.
No, and we have actually made measurements of urea in sea ice as well, and sometimes it gets to 100 times more than you normally find in the system, but never to the amount that ammonium is accumulating in the sea ice. I've not read of any nitrogen fixation. I mean, in the marine systems, nitrogen fixation organisms are the cyanobacteria, which used to be called the blue-green algae. In the Antarctic waters, they don't play much of a role. They do play a role in milk ponds up in the Arctic. So there's a lot of blue-green algae, so cyanobacteria, up in the Arctic milk ponds, but in the Antarctic. But I mean, we have to remember, so, obviously carbon, oxygen, and nitrogen is important, but of course phosphorus is important, and so is sulfur, and all the way down this kind of ever-decreasing spiral. And this is the view that, we always have to keep in mind. If you don't have any sulfur in the system, you're not going to get any growth, or you don't have any phosphorus in the system, you're not going to have any growth as well. And that's why, iron has become a big thing in the Antarctic. You throw a load of iron into patches in the Southern Ocean, and you get these blooms of algae because iron is limiting to algal growth. It's only needed in very, very trace amounts, but it still is a vital limiting factor. So you know, we get hooked up on nitrogen and CO2 and oxygen, and to a lesser extent phosphorus in marine systems. But of course, you know, we do have to look at trace metals as well. Okay, we're nearly finished with the definitions. Dissolved organic matter has become basically the dissolved organic matter is what is fueling the microbial system, the bacterial system within marine systems. If you filter some water through a 1 micron filter, it will contain all of these crustaceans and zooplankton. It will keep out most of the algae, but a lot of bacteria will go through the pores of the 1 micron tray. So if you then use a 0.45 micron, most of the bacteria will be on it, but some will get through. If you see, the point what I'm trying to make is the elimination between the dissolved and the particulate phase is a very difficult one to do because it's defined operationally by your filter size. You know, so in the early 60s, 1 micron was the definition. Now 0.2 micron is used to define it. It's just a very nebulous kind of idea that for some reason marine biochemists haven't really got a handle. But basically, a 0.2 micron filter keeps out all bacteria, but most viruses go through, and some strange things. So what is dissolved and what is particulate is a bit odd. But within a marine system, and in its most varied form, you have this phytoplankton and eutrophic network that are uptaking nutrients and carbon and then putting them back into the system. You obviously have things that are particulate organic matter, and you have things that are dissolved as well. They're excreting things, as I'm going to show you as well in a minute. But the important thing is, especially in a system like the Arctic, where you have a huge amount of river input into the system. In the Antarctic, it's not so important. You have a large pterogenic input of particulate material and dissolved material as well. So it is important that in certain parts of the world that it's not just the autochthonous organic matter that is considered, but also the allochthonous, that comes from the outside, is actually considered as well. But if we look at organisms producing stuff and what happens, so phytoplankton, copepods, fish, whales, whatever, they all exude things or produce feces or whatever, or excrete stuff. And this all goes into this kind of extracellular organic pool, which is then what the bacteria are breaking down for their growth. When an organism dies, it's going to eventually break open and release all of its cell contents into the water. So, and when an organism eats another organism, quite often it's not done in a very, very tidy or uniform way. Sloppy feeding is a big field where, you know, a copepod may crack open a diatom, may suck up some of the juices, but most of them go off into the water surrounding it. So all of this is leading to this dissolved pool of organic matter that the bacteria are then able to utilize to break down. I noticed this has happened. I don't know what's happened here. Basically, you have phytoplankton that take CO2, sunlight, and nutrients. They photosynthesize, and then they're the food for a whole host of other organisms, up to whales or humans, whatever you want. But everything is excreting this organic matter into this pool. And this is what the bacteria are utilizing for the bacterial growth. But a consequence of the growth or the conversion of the dissolved organic matter into bacterial biomass is that a lot of nitrogen and phosphorus and trace minerals and everything else are released back into the water that are then used by phytoplankton for growth. So basically, until this part of the system was discovered, this is all that people thought happened in marine systems. They didn't really know where this was coming from. And that's in the last 30 years. And this is another depiction of it. And this is what leads to the complexity of the cycles So this is a rough appreciation of the nitrogen cycle within marine waters. Well, basically, this little cycling bit here, well, actually no, this bit here is all reflected in this kind of subplot that is going on here. Now again, so it's becoming, just for nitrogen, it becomes a very complex story. But the thing is that this complexity is all mediated through the biology. If you like, the biology is acting as the catalyst for all of these complex reactions. The same with the sulfur cycle as well. There's a complex set of interactions that are going on, even kind of going up to cloud nuclearization. But basically, it's all being kind of mediated or catalyzed by the biology. If you ever want to really upset a first-year group of students that have come to study marine biology because they're interested in whales and dolphins and mammals, the charismatics, you just say, well, the biology is just a catalyst for the chemistry to go on, and you just see their faces drop. So basically, I mean, but it's true. I mean, this is what we're beginning to discover is going on within the ice as well. But I think it's time for questions or a break at this point.
Thanks, David. Questions, please.
But it's from the very beginning when you showed pictures of these diatoms and some of the experiments that people did in Bremerhaven. I saw a number of a Newton per square millimeter, which was von Mises stress. So they must have the critical bending stress before one breaks. Has that been measured?
They have done those measurements, as far as I know.
Okay. So is there an observation of a diatom being frozen completely into the ice? That's the first question. Second question is there a measurement of osmotic pressure?
The measurements of osmotic pressure have been done, but the measurements of diatoms being frozen, no, they haven't. Because I mean, the only pictures that exist of diatoms being encased in ice are those ones, if you like, that Christopher Krenz, isn't it?
Right.
But that's all in rather warm sea ice,
right,
if you like, where he's able to take photographs. So you know, the pictures of diatoms in -20 sea ice don't exist as such.
Okay. It's just Harold measures what happens when ice gets near surface, and so do we. And I just calculated the thermomolecular pressure for that sort of circumstance for silicate, and it's equal to the same number you gave me. So I was just wondering if there was, you know, besides the crustacean's teeth evolving to be strong enough to crush one, I was just wondering if there was more mechanical measurements made on these things.
Well, no, that would be interesting, because I mean, the group that did that work has now been kind of seduced by VW and BMW into making wheel hubcaps and things like that. So they're getting serious cash from their work. Whereas, and that group, as far as I know, haven't ever kind of linked up with the sea ice, you know, Gerd Dickmann or this sea ice people in the RB to do that, because that would be the obvious thing to look at. And as far as I know, that's certainly never been published.
Right.
They never told me about it. But that would be interesting to do.
What about the heat that is released during photosynthesis? How much is that actually?
Yeah, this has been there was a modelling exercise that was done by Richard Zieber a little while ago, and what well, it wasn't so much the heat that's generated during photosynthesis, because that's really quite low. But what the interesting thing that they did is they were modelling the algal layers on the bottom of an ice floe acting as a heat absorber, and therefore as a black body, and what was the role of that in melting the ice. And to be quite honest, the numbers are they just weren't convincing. I don't think it actually has a role to play in nature. I mean, intuitively we all thought that, you know, that was a real answer, but I don't think it is. The heat generator is not.
But you did mention it. So
I did mention it. So I mean, ultimately, yes, but a lot of that heat energy is
yes.
I mean, these diagrams of the cycle where, you know, the bacteria are doing their things with the dissolved organic matter, does it mean that in a confined system, or a more confined system, say the ice rather than the ocean, that there is particular role for bacteria that is, I mean, is the concentration of bacteria in the ice different than somewhere else in the ocean just
No.
because, no?
Yes, no, it is.
Yeah.
No, it absolutely
Yeah.
is. And that's one of the points that I want to make, and that it's worth making here, is that this, when you concentrate that biology within the ice, it's a whole completely different system. So at the moment we try and imagine what is going on in the ice by what we know from the open waters. My guess is that's completely wrong, because the whole of the interactions, with bacteria, you're bringing algal cells that will be meters apart, well, let's say centimeters apart from their neighboring algal cells in nature, that you're bringing them, within micrometer scales. So all of the gas exchange processes are all completely different, and I think we've got a lot to learn about that.
So follow on that question then. I mean, are there, in the ocean, is there a particular rate limiting part of that cycle that's the sort of the bottleneck, and is it different is there a bottleneck, and does
The bottleneck in the conversion of DOM to nutrients, that kind of cycle
Yeah.
we're talking about. The bottleneck is probably bacterial growth rate, if you're looking at a rate limiting step, of which of course in the ice is very dependent on temperature. So it's dependent on the amount of organic matter you give it. But when we measure organic matter in the ice, they're very much higher concentrations than ever you see in the open waters. So my guess is for the bacteria in that system, it would be temperature. Yeah.
You talked about the depth of the photosynthesis, of in the sense of roughly 50 meters or so in open water. How deep are we talking in ice? It must really be the surface.
No. I mean,
How far does that go in a sense?
yeah. So light. Well, it can't be just the surface, because we see that you take any ice flows just about anywhere that you go to, you will see some biological activity. Ice flows that are 3 meters thick still have, with no snow on top, still have algae growing that are photosynthesizing. They're just very, very low light adapted. So they tend to have an awful lot of pigment per cell to actually enable them to grow still at very low
We're
light.
still talking about a few meters.
But you're talking so you're talking about 4 or 5 meters.
Yeah.
And that's when there's no snow.
Yeah.
You put even a thin layer of snow on, and then it's basically black under ice. So.
I was just going to say, the ice has got a Beer's extinction coefficient of about 4 meters, spectrally averaged. So it can go pretty far, but it brings up the question of what might happen elsewhere, because you've got, if you're talking only about photosynthetic life, these processes, then you've got a competition between how deep you can get the light and how cold it is.
Yeah.
And you can't get the light that deep
No.
in ice.
Well, how are the guys moving the guys? Do we know how fast do they move?
How fast? As one of the steps of experiment, I don't know how fast they move. These diatoms, they tend to be motile, although they don't tend to be. A lot of the species are actually motile. So when they were first identified by the gentleman scientist that first looked under the microscope and saw them, they actually called them animalcules, because they actually saw them moving. They move on a polysaccharide slime trail, so they can actually move up and down inside brine channels. Bacteria have been shown by Jodie Deming and her group to still be motile, well, let's say down to minus 15 degrees. So ice that is at minus 15, the bacteria are still motile-ish. But certainly down to minus 8 degrees, I think motility is not an issue for many of the organisms that are living there. Space is the thing that's limiting them.
The spectral properties were rather amazing. That at great depth, it's a very, very narrow spectral range that gets transmitted. And that was the amplitude actually normalized to?
No, that wasn't.
So really, within the right spectral line, you have hardly any absorption.
Yeah, that's
over
right.
great depth mouth.
Was it normalized?
It was not normalized.
It was not normalized.
Absolutely.
But I mean, the peak is little similar to those.
Yeah, I mean, it was kind of, this was from a textbook, it was kind of idealizing situations.
So the equivalent behavior in Iceland.
Yeah, I got to admit, I mean, if you want to know about the optics, I suppose you should read Don Perovich's work on this kind of stuff. So Perovich is the guy who's done most of these optical properties. But there are groups out there that have measured UV light, which, again, I'm amazed at. You know, under 1 meter thick ice, they still measure some UV getting down to the bottom of a 1 meter thick ice flow. So, which surprises me, but there you are. They've measured it.
So once you go down to the dark region, so to speak, in ice, do you see a transition into other forms of organisms?
No,
Not.
It doesn't tend to be. I mean, most of my experience, from what I know of literature, is the problem with ice is that, you know, if it was just an ice flow that was 6 meters thick and it just kind of went around the ocean as a 6 meter thick ice flow, that would be fine, and it would be permanently dark. But what will happen to that ice flow is it will be broken up, it will be jumbled around, turned upside down, or whatever. So when you look at the biology that is in the biological history of an ice flow, it's very murky for knowing where that biology is grown. You know, probably what you find at the bottom of an ice flow may not necessarily have always been the bottom of that ice flow.
Well, I mean, in the summer it melts from the bottom and it melts from the top.
Yeah.
In the winter it starts to grow from the bottom while it's still melting from the top in the fall. And what Jerome showed was that probably it's not, I mean, I don't know the answer to this question, but once we have these channels forming, the major conduits through which there's transport, nutrient transport, are localized. And so you would imagine, you know, life would congregate around these places because then it doesn't have to go anywhere. Some type of life.
Yeah, no, exactly. So, and the studies where you actually follow a lump of ice and you know from colonization through to melt at the end of the period, they are incredibly rare. People just don't do them because they don't have access to the ice, particularly. There are a few out there that don't.
It's harder on Europa, though.
Yeah. Nobody's asked, well, how does it link in with Europa? I'm just doing what I was asked to.
So, should we have an hour break? Then we continue? Thanks.